Every parent is nervous when a new baby is on the way. There are so many questions that run through the parent's head as they must prepare for the baby's essential needs. In the 21st century, a baby's crib will consist of many colorful toys and stuffed animals. Parents put these toys inside the crib with the baby in hopes to enhance the baby's brain at an early age. This idea comes as a question whether certain experiences stimulate the brain has been the topic of research. Several scientists have all conducted similar experiments in hopes to see the relations between the effect of experiences on early brain development. In 1785, Vincenzo Mallarcani conducted an experiment observing the brain's development in dogs from the same litter and birds from the same batch of eggs. He would extensively train one dog while the other would not be but was equally taken care of. He discovered later in the autopsies of dogs that the ones that were trained extensively over a long period of time had more folds and fissures in their brains. However, nobody can claim that more folds and fissures meant that the brain was more complex. By the 1960s, improved technology allowed the scientists to measure the brain's complexity more precisely. Mark Roentgen and Marion Diamond redesigned and continued Mallarcani's experiment in an ambitious series of 16 experiments over 10 years of the effect of experiences on the brain. The hypothesis of this experiment is as follows. If a child is placed in a stimulated learning environment, then the effect of experiences will impact the brain.
3 male rats were chosen from each litter. They were then randomly assigned to one of 3 conditions. One rat remained in the laboratory cage with the rest of the colony, another was assigned to what Rosenzweig termed the enriched environment cage, and the third was assigned to the impoverished cage. Remember, 12 rats were placed in each of these conditions for each of the 16 experiments. The 3 different environments are described as follows. The standard laboratory colony cage contained several rats in an adequate space with food and water always available. The impoverished environment was a slightly smaller cage isolated in a separate room in which the rat was placed alone with adequate food and water. The enriched environment was virtually a rat's Disneyland or a place of total bliss. 6 to 8 rats lived in a large cage furnished with a variety of objects with which they could play. A new set of playthings drawn out of a pool of 25 objects was placed in the cage every day. The rats were allowed to live in these different environments for various periods of time ranging from 4 to 10 weeks. Following this differential treatment period, the experimental rodents were examined to determine if any differences had risen in brain development. To be sure that no experimental bias would occur, the examinations were done in random order by code number so that the person doing the autopsy would not know in which condition the rat was raised. The rats' brains were then measured, weighed, and analyzed to determine the amount of cell growth and levels of neurotransmitter activity.
Results indicated that the brains of the enriched rats were indeed different from those of the impoverished rats in many ways. The cerebral cortex of the enriched rats was significantly heavier and thicker. Also, greater activity of the nervous system enzyme acetylcholinesterase was found in the brain tissue of the rats with the enriched experience. Although no significant differences were found between 2 groups of the number of brain cells, the enriched environments produced larger neurons. Related to this was the finding that the ratio of RNA to DNA was greater for the enriched rats, which implied that a higher level of chemical activity had taken place in the enriched rat's brain. Also, under great magnification using the electron microscope, the researchers found that the synapses of the enriched rats' brains were 50% larger than those of the impoverished rats, potentially allowing for increased brain activity.
Why is this so important? Well, this study proves that the learning environment does play a role in a more stimulated brain.